Sodium Dodecyl sulfate, sodium lauryl sulfate or sodium lauryl sulfate is an organic compound with the formula CH3, CH2, 11OSO3 Na. It is an anionic surfactant used in many cleaning and hygiene products. The salt is of an organosulfate consisting of a 12-carbon tail attached to a sulfate group, giving the material the amphiphilic properties required of a detergent. Derived from inexpensive coconut and palm oils, it is a common component of many domestic cleaning products. Sodium coco-sulfate is essentially the same compound, but made from less purified coconut oil. Production: SDS is synthesized by treating lauryl alcohol with sulfur trioxide gas, oleum, or chlorosulfuric acid to produce hydrogen lauryl sulfate. The industrially practiced method typically uses sulfur trioxide gas. The resulting product is then neutralized through the addition of sodium hydroxide or sodium carbonate. Lauryl alcohol is in turn usually derived from either coconut or palm kernel oil by hydrolysis, which liberates their fatty acids, followed by hydrogenation. Due to this synthesis method, commercial samples of SDS are often a mixture of other alkyl sulfates, Dodecyl sulfate being the main component. SDS is available commercially in powder and pellet forms. It seems the pellet form dissolves faster than the powder form in water. Applications: SDS is mainly used in detergents for laundry with many cleaning applications. SDS is a highly effective surfactant and is used in any task requiring the removal of oily stains and residues. For example, it is found in higher concentrations with industrial products including engine degreasers, floor cleaners, and car wash soaps. It is found in toothpastes, shampoos, shaving creams, and bubble bath formulations in part for its thickening effect and its ability to create a lather. Pepsodent toothpaste at one time used the name Derived for its sodium lauryl sulfate ingredient. Laboratory applications: it can be used to aid in lysing cells during DNA extraction and for unraveling proteins in SDS page. Sodium lauryl sulfate, in science referred to as sodium dodecyl sulfate or duponol, is commonly used in preparing proteins for electrophoresis in the SDS page technique. This compound works by disrupting non-covalent bonds in the proteins, denaturing them, and causing the molecules to lose their native shape. This new negative charge is significantly greater than the original charge of that protein. The electrostatic repulsion that is created by binding of SDS causes proteins to unfold into a rod-like shape, thereby eliminating differences in shape as a factor for separation in the gel. Sodium lauryl sulfate is probably the most researched anionic surfactant compound. Like all detergent surfactants, sodium lauryl sulfate removes oils from the skin, and can cause skin and eye irritation. The critical micelle concentration in pure water at 25 degrees Celsius is 8.2 mm, and the aggregation number at this concentration is usually considered to be about 62. The micelle ionization fraction is around 0.3. Aqueous solutions of SDS are also popular for dispersing or suspending nanotubes. Sodium lauryl sulfate is also used in the analysis of hemoglobin. The hydrophobic group of SLS acts upon the globin subunit, causing a conformational change. The hydrophilic group of SLS then binds with the oxidized iron subunit, producing a stable reaction product which can then be analyzed, giving a hemoglobin value which is used as part of a complete blood count. Sodium dodecyl sulfate is also used to clear brain tissue, by removing lipids responsible for light scattering. Biological tissues treated by sodium dodecyl sulfate are rendered quasi-transparent in optical microscopy, enabling to see through them. Niche uses: biocide, SDS represent a potentially effective topical microbicide, which can also inhibit and possibly prevent infection by various enveloped and non-enveloped viruses such as the herpes simplex viruses, HIV, and the Semliki forest virus. Shark repellent: evidence suggests that surfactants such as sodium lauryl sulfate can act as a shark repellent at concentrations on the order of 100 parts per million. However, this does not meet the desired cloud deterrence level of 0.1 parts per million. Pesticide: some species of flies, ants, and moths are attracted to sodium lauryl sulfate, even in small amounts, and are then killed by it. It is the main active ingredient in most store-bought fruit fly killers. Fruit flies are more commonly attracted to it, however, if the chemical is in a sweet-smelling soap or detergent. Taste alteration. Sodium lauryl sulfate temporarily diminishes perception of sweetness, an effect commonly observed after recent use of toothpaste containing this ingredient. Toxicology, carcinogenicity. SDS is not carcinogenic when either applied directly to skin or consumed. A review of the scientific literature stated SDS was negative in an Ames test, a gene mutation and sister chromatid exchange test in mammalian cells, as well as in an in vivo micronucleus assay in mice. The negative results from in vitro and in vivo studies indicate SDS does not interact with DNA. The same review also stated in the only carcinogenicity study available, SDS was not carcinogenic in beagle dogs, though the short study duration and limited details provided limit the significance that can be attached to the data. Sensitivity. It has been shown to irritate the skin of the face, with prolonged and constant exposure in young adults. SDS may worsen skin problems in individuals with chronic skin hypersensitivity, with some people being affected more than others. In animal studies, SDS appears to cause skin and eye irritation. SDS can contact the face from aerosolized dust during SDS reparation for laboratory purposes. To prevent contact with face and eyes, a dust-free epilethly form of SDS is available which essentially eliminates SDS dust inhalation and contact with the face. Aphthous ulcers. There have been several studies on how SDS in toothpaste affects the recurrence of aphthous ulcers, commonly referred to in some countries as canker sores or white sores. The results of these studies have been inconsistent. In 1994, a preliminary crossover study showed patients had a significantly higher number of aphthous ulcers after using SLS-containing toothpaste, compared with an SLS-free toothpaste. A follow-up double-blind crossover study in 1996 further supported these results, as did a separate study in 1997. However, a double-blind crossover study published in 1999 failed to find any statistically significant difference. A double-blind crossover study in 2012 also failed to find a significant difference in number of ulcers, but did find a significant difference in ulcer duration and pain scores. According to the 2012 study, patients using an SLS-free toothpaste experienced faster healing of ulcers and less ulcer-related pain on average than patients using SLS-containing toothpaste. Rembrandt Gentle White Toothpaste, which does not contain SLS, was formerly sold by Johnson & Johnson. Interaction with fluoride. Some studies have suggested that SLS in toothpaste may decrease the effectiveness of fluoride at preventing dental caries. This may be due to SLS interacting with the deposition of fluoride on tooth enamel. See also, ammonium lauryl sulfate, potassium lauryl sulfate, sodium myrith sulfate. References